This is part 2 of the lecture notes for chapter 9 on the cell cycle. I was trying to. Sorry, I'm trying to make the screen a little bit bigger so you can see the actual slide number. I don't know if I can do it or not. Well, I can pull it down. Okay, so that you can see down at the bottom that this is slide number 20. Anyway, I don't guess it's that big of a deal, but sorry, I'm worrying about things that I don't need to be worrying about. We are on slide number 20, and we are still in the section where we're learning about chromosomes. Okay, so when chromosomes duplicate, we also call that replication. But basically, a single chromosome makes a copy of itself, and the single chromosome is on the left, and you can see that when it copies itself, now you have two sister chromatids. The region where they're attached is called the centromere, and then the little orange-yellow structure that the spindle fibers are going to attach to during prometaphase, during the stage called prometaphase. That structure is called the kinetochore. Okay, so back to the notes, we would say, so during interphase, the cell is preparing to divide. Not only is the DNA or chromosomes duplicated, but also the organelles. So organelles are duplicated. At the end of the S stage, you're going to have two sister chromatids at the end of the S stage, two strands of genetically identical chromosomes. They're going to be attached at a point called the centromere, and then the structure on the centromere where the spindle fibers will attach is called the kinetochore. Okay, so that's going to be something you have to make sure you understand the difference. The sister chromatids are attached to one another at the centromere, but the kinetochore is the structure that the spindle fibers are going to attach to. Okay. Now, okay, I will tell you there is a structure called the centrosome in eukaryotic cells. It is in animal cells, it is called the microtubule organizing center. The microtubule organizing center. The centrosome is replicated, and in animal cells, the centrosome also contains these two barrel-shaped structures called centrioles. But plant cells don't have them. Plant cells have a centrosome, which is a area that helps organize the spindle fibers. Microtubules, remember, are the type of fiber, the type of protein fiber that form the spindle fibers. And so down here it says the centrosome organizes the mitotic spindle, and the spindle contains many fibers. Those fibers are microtubules. And what the microtubules do is they allow the cell to change shape for cell division, and those spindle fibers are going to attach to the chromosomes, and they're going to pull the sister chromatids apart whenever we get to the phase called anaphase. Now, we are going to cover all of the phases of mitosis, and they're listed here. There's prophase, there's prometaphase, then metaphase, anaphase, and telophase. And you can see images here of the stages. You can see what prophase looks like, what prometaphase looks like, metaphase, anaphase, and telophase, and what those stages look like under the microscope for animal cells, which are up at the top, and then for plant cells, which are down here at the bottom. So we're going to start with prophase and make sure that you know all the events that happen in prophase. Now this is going to seem like a lot to have to learn. So let me just tell you, for all of these phases of mitosis, the only one that's really going to be hard to learn is prophase. So let's go ahead and cover prophase. During prophase, the chromatin condenses into chromosomes, and you can see those chromosomes under the microscope. So that's the number one thing that happens in prophase is the chromatin, which was very spread out and diffuse during interphase. Now it condenses and forms those linear rod-shaped chromosomes. And they're actually X shapes because they're the two sister chromatids attached at the centromere kind of looks like an X, the letter X. So each chromosome has two sister chromatids held together at the centromere. That's one thing that happens in prophase. This will be the first event that happens in prophase is chromatin condenses and forms chromosomes. Another thing that happens is the nucleolus, and I don't know if you remember, but the nucleus contains the DNA, contains the chromatin, and the nucleolus is a structure inside the nucleus that forms the ribosomes. So the nucleolus disappears and also the nuclear envelope. So basically, the nuclear envelope begins to fragment and dissolve into the cytoplasm. So if we just say the nucleus disappears, that kind of covers both of these events. So if number 2, we just say the contents of the nucleus disappear, that will cover those events. I'm trying to keep things very simple. Okay, then the spindle begins to form. It begins to assemble or form. So the spindle appears. The nucleus disappears, the spindle appears. And the centrosomes begin to move away from each other toward opposite ends of the cell. And that would be the fourth event that happens in prophase. All right, in prometaphase, you only need to know one thing, and that is that the spindle fibers connect to the kinetochores of the sister chromatids. So prometaphase, your spindle fibers, it doesn't say it like that, but spindle fibers, since it's not in here, the spindle fibers or spindle microtubules attach to the kinetochores of each pair of sister chromatids. And then in metaphase, the spindle fibers organize the chromosomes to where the sister chromatids, they're pulled until they're lined up at the metaphase plate, which is the middle of the cell. So they align across the equatorial plane of the cell at the metaphase plate. In other words, the chromosomes line up at the middle of the cell. And then in anaphase, these spindle fibers begin to shorten, and as they shorten, they pull the sister chromatids apart. They separate the sister chromatids. That's anaphase. Sister chromatids separate. And then telophase has a lot of events in it as well, but they're going to be opposite to prophase. So if we go back to prophase, chromatin condenses into chromosomes. So now we're going to go back, and in telophase, the chromosomes become diffused chromatin once again. So that would be the first event that happens in telophase. The second event is going to be opposite, where the nucleus disappears. Now the nucleus is going to reappear. It's going to reassemble. We'll say the nucleus reforms, and that means everything inside the nucleus. That means the nucleolus as well. So that is the second event. The third event should be that the spindle is going to disassemble. So the spindle disappears or disassembles. That would be the third event. And then the fourth event says that the centrosomes move away from each other. And it doesn't really say anything about them. So just learn these three events in telophase: that the nucleus reforms, chromosomes become chromatin once again, and the spindle disappears. Okay. Um, And you can see here in early prophase, you can see that the chromatin has condensed and formed into chromosomes, or it's starting to. The nuclear envelope is beginning to break down. The nucleolus is beginning to disappear. The centrosomes are forming, and the asters are going to be the spindle fibers kind of radiating out away from the centrosome and the centrioles. In prophase, you can see in the illustration that your chromosomes are visible, the nucleus has disappeared, the centrosomes are moving to opposite ends of the cell, and the spindle is forming. The spindle fibers are forming, so they are assembling. In prometaphase, the spindle fibers are attaching to the kinetochores of the chromosomes. And once they attach, they're going to be able to arrange them so that they line up in metaphase. So the spindle fibers help the chromosomes line up at the metaphase plate during metaphase. In anaphase, the spindle fibers they separate the sister chromatids. And in telophase, the sister chromatids arrive at opposite ends of the cell. The cell begins to undergo cytokinesis. It begins to pinch in half. And you can see the nuclear envelope reforming around the two sets of chromosomes. And you can see that the spindle has disappeared. Now, in plant cells, cytokinesis in plant and animal cells is different. Mitosis is the same in plant and animal cells, but cytokinesis is different. Cytokinesis is the actual pinching in half of the cell. It's the division of the cytoplasm. And in animal cells, this happens by the formation of a cleavage furrow. And a cleavage furrow forms by a contractile ring of actin filaments that literally divide the cell in half until you have two cells with, surrounded by two separate plasma membranes. In plant cells, cytokinesis begins with the formation of a cell plate. And a cell plate is formed by many small vesicles that are made by the Golgi apparatus. And those vesicles fuse into one very, very thin vesicle, and that's what becomes the cell plate. And later on, the cell plate becomes the cell wall in between the two cells. So here you see an illustration of the cleavage furrow that forms during cytokinesis of animal cells. And you see an illustration of, well, actually, this is an electron microscope image of the vesicles that form the cell plate in a plant cell. So plant cells form a cell plate and animal cells form a cleavage furrow. Functions of mitosis. Mitosis functions in growth and repair of organisms. When the fertilized egg divides and becomes an embryo, and then when the embryo becomes a fetus, and then when the child becomes an adult. In other words, as the organism grows larger and its single fertilized egg cell, which is called a zygote, divides into trillions of cells as the organism grows into an adult. And then the other thing is repair. So when a cut heals or a broken bone mends, you're going to have mitosis of cells that are needed in the repair process. All right, stem cells are cells that can divide and produce new cells. In therapeutic cloning, that is using adult stem cells or embryonic stem cells. Embryonic stem cells can be used for reproductive cloning, which is the production of a new individual, but adult stem cells cannot.